Dear student friends, I am Dr. Godbole Mahendra. Again, welcoming you all in my today's presentation, which is based on the biotechnology. The biotechnology is very important branch of the biology, where the genetic engineering is playing very crucial role for to modify the phenotypes, and the genetic engineering is very important to crop the desirable sequences of any species or any maybe microbes, and that technology is very important for the changing the phenotypes or the second filial generations. In this biotechnology, the endonuclease or the restricted enzymes is very important for to cropping the pre-existing DNA sequences. What happening in earlier days, the farmers, especially the Indian farmers, were not able to producing good quality of the yield of the cotton. It is because of many those insects were destroying the crops, especially the ball of the cotton. But nowadays, after the development of genetically engineered BT cotton means that is Bacillus thuringiensis species has played a important role to modify the morphological feature and the chemical characters of that particular cotton, so that the cotton may restrict the insects and the cotton of the ball may use good quality of the cotton. That is the application of the technology. Biotechnology playing a very important role in industries for the development of the antibiotics, for example, and the formation of the different enzymes, or the different kinds of the medicine can be synthesized by those biotechnologically or genetically engineered varieties. According to the René Descartes, as a French philosopher and the mathematician as well as a biologist, what he said that in the he has given his service for the good or the well-being of the human being, and he has worked in the natural sciences. He has directed to develop the technologies. The Rain has developed the technology and which he has added to create the comfort life of the human being, and he also value the human life, because as per the genetic engineering, the human life has become more comfortable, and the understanding of the natural phenomena become the anthropocentric. Nowadays, the physics, chemistry gives rise to engineering technologies and the industries which allows the work for the human comfort and the welfare. Means combinedly, either the material science or the biological science, both are going hand in a hand, and their amalgamated technology is going to making our life more comfortable. The major utility of this biological world is as a source of the food. And what is the ultimate aim of the biotechnology? To yield the production of any crops at the greater extent, so that the increasing populations request or the quench may be thirst. Their thirst may be quenched. The major utility of this biological world is a source of the food. Biotechnology is a 20th century's offshoot of the modern biology, changed our daily life as its products brought the qualitative improvement in our health, food production, the basic principles of the underlying biotechnological processes, and some applications are highlighted and discussed in this unit. In this unit, I am going to explaining about the principles of the biotechnology and the different kinds of the applications of the biotechnology. What is biotechnology? The EFB means the European Federation of the Biotechnology has been defined What is the biotechnology? Biotechnology deals with the techniques of using the live organisms or the enzymes. In biotechnology, either the life of any organism, living microorganism, especially, let us take an example, the E. coli bacteria. Earlier, the human has used, synthesized, the Toronto University scientist has synthesized the E. coli bacteria's enzyme for to control the different diseases. Now the biotechnology has defined by the European Union or the Federation of the Biotechnologists from Europe, the integration of the natural sciences. The biotechnology is an integration of the natural sciences, the desired and the pre-existing species amalgamation or integration of the science, organisms, cells, part of thereof. Any organism, its cell or the part of the cell or the tissue or the molecule, and it must be analogous. It may be same. It must be analogous for the production and the services. In biotechnology, any organism, any species cell or the part of the cell or the tissue can be used for this development of this technology. What are the principles of the biotechnology? There are the two principles of the biotechnology. Those adoption of these two important principles of the biotechnology, one is a genetic engineering. The first one is the genetic engineering and another one is the bioprocess engineering. What is genetic engineering? In this technology, the chemistry of the genetic material as a DNA which is situated in the nucleoplasm of the cell as a DNA and RNA to introduce these into the host organism. The techniques, it is a chemistry is involved in this technology. The techniques to alter the chemistry of the genetic material. The genetic material is used or to introduce these into the host genetic host organisms and thus the changes phenotypes of the host organism. Whichever the host organism is supposed to be under consideration to change, that host may be changed by certain techniques by the insertion of the desired sequences of the DNA is possible and therefore the new species may be developed. Let us take an example. The earlier crop as a cotton was prone to the different insect, but the genetically engineered variety as a BT cotton, Bacillus thuringiensis species, was able to stop the insects and it was giving a more crop, more yield. Now the next principle of the biotechnology is bioprocess engineering. What is bioprocess engineering? It is a laboratory based activities, maintenance of the sterile or the microbial contamination free. Wherever the biologist is going to working in a laboratory, that condition has to be sterile. There must be, the biologist has to avoid any kind of the contamination so that the phenotypes may not alter any kind of the morphological feature. And this is a contamination free environment, ambience in the chemical engineering processes to enable growth of only the desired microbe. If we are going to inserting certain desirable sequences of the DNA in a host so that the contamination has to be avoided for the proper growth of the phenotypes of the next generation. In eukaryotic cell, in the large quantities for the manufacture of the biotechnological products like the antibiotics, vaccines and enzyme. Nowadays, the biotechnology is most important science or engineering. It is widely used for the antibiotics, vaccines and enzymes. These are the outcomes of the biotechnology. And what are the basic steps? Which basic steps are supposed to be involved in the engineering, especially the genetic engineering? The identification of the DNA with the desired genes. Every DNA is having a double strand, and every the DNA segment is having a certain kind of the sequence, as a every nucleotides also, and those sequences may possess a certain kinds of the character, and those character can be desirable character. Okay, and the next step, for after the identification of the DNA with the desirable genes, there are many genes on the chromosome or on the DNA, and after the identification of the desired DNA, the introduction or introduction of the identified DNA into the host cell. For example, this is a species which having a resistance. For example, here in this DNA, there is one codon, and which is supposed to be having a capacity to sustain in a diverse situation. Another sequence may be for the earlier maturation. So this desirable character may be introduced to the host cell, then maintenance of the introduced DNA in a host and transfer of the DNA. The maintenance has to be done after the insertion or introduction of the desirable DNA to the host cell, and then the process maybe goes on. Now, what are the molecular tools or the restriction enzymes? The restriction enzymes can be synthesized or can be acquired by the different vectors, and those synthesizers, those molecules or those restriction enzymes may be act as a seizure. We know how the seizure can separate the any desirable part of any material. Likewise, here also, this scissor is supposed to be cropping certain segment of the DNA or the desirable DNA's part. Every DNA's, that is adenosine, thymine, cytosine, those are the sequences on the every stair-like part or the appearance of the DNA, and each segment as a gene is having a unique feature, and those desirable character may be inserted to the host cell, and this is supposed to be inserting cropping of the existing pre-existing or the selective species DNA, and you know it is going to cropping through the scissor by certain enzymes, and this is supposed to be the recombinant DNA technology. This is a desirable and this is a host cell, and the ultimate product will be the recombination of the DNA as a Bacillus thuringium bt cotton and pre-existing earlier normal cotton. Now, the isolation of the DNA is possible by means of the restricted enzymes, and the cutting of the DNA or through the restriction enzyme, it may be ligating or joining the DNA. DNA may be joined. For example, the human E. coli bacteria was used for synthesizing of the certain enzymes. Now, there is introducing this host cell, and after the host cells, the selection of the screening will be takes place, and later on there may be the formation and the production of the desirable product. This desirable product may have the characters, both type of the characters. Okay, the pre-existing varieties character, those character were selected, and the host cells character, both the characters will be combined in the productions of the desirable species. These two principles, as a genetic engineering and the maintenance of the sterile environment means the contamination free environment is very important. Contamination free. Contamination has to be avoided so that the proper growth of the phenotypes may be developed. These are the application of the biotechnology, and the new product is available in our market. For example, in agriculture, the genetically modified crops, GM crops, medicine, what kind of the products are available in a market? The vaccines and insulins, those are also the genetically modified product. Industry, enzymes and the biofuel is also genetically modified. In environment, that is bioremediation. Bioremediation is also important. In food technology, fermentation and fermented food is also available in our market. Hence, the biotechnology is going to changing the lifestyle, and the lifestyle of the human is goes on increasing, and the lifespan is also increasing, and comfortability is also increasing by the application of the biotechnology. The top 10 genetically modified food is here. I am going to show you these 10 varieties. That is, corn is a genetically modified variety. Earlier, the corn's production was very limited because of the insects, because of the corn species was not able to sustain in a very least precipitation, but the genetically desired and modified varieties may sustain in a very limited precipitation and in any kind of the diverse climatic condition. Therefore, this is very important. Now, the soya is also used for the protein and other development. Cotton is also the genetically modified cotton, or the BT cotton is also giving it is very important for the industrial growth, for the textile industry, for that cloth industry. The cotton is also important. Papaya is also important. It is also increasing day by day its production by means of the genetically modification. Rice is also important. That is a fifth type of the genetically modified variety. Some American biologists or a few American botanist has cropped Indian certain Indian rice DNA, and it has amalgamated with their own variety, and they got the patent on the rice. And the other, the development of the beautification, that is canola or the papseed, is also genetically modified. And in Indian food, at extreme north to south, we will see the potato is mainly used, and that is also genetically modified, means it grows at a larger and a great extent. Then tomato is also important. It is also genetically modified. And dairy products, or the peanut is also giving a very good yield, and it is going to supplying for the increasing population. Not only increasing population, but our genetically modified varieties, we are going to exporting to the foreign countries. In the Middle East, the different the Tahsil of the Nanded is going to sending, and those are going to exporting, just like many genetically modified varieties, including this potato and tomato, as well as the banana is also exported by different parts of the India. Even the Nashik is also going to exporting the different parthenogenic food to the other countries. These are the biotechnology, and it is not just a subject, it is a evolution, it is a service to the human being. Biotechnology, it is not it is not a mere science, it is a evolution. These are the steps which are very important. For example, the that is a intestinal cell. The intestinal cell was also amalgamated with each other and form the different microbial combination. Biotechnology is a multidisciplinary science where the physics and chemistry and biology and different, the material science as well as the natural science is involved in this. And that science uses for a living organism, cells, enzymes, biological systems to develop the products and the technologies for the benefits of the human being. What is the ultimate aim of the biotechnology? Ultimate of the biotechnology is to increase the yield so that the increased productions may reaches to all corner of the country. Genetic engineering is also earlier I have been discussed how this cropping of the gene or the genome or the nucleotide is possible, and this is a dissolution of the protein synthesis, and different kinds of the segmentations can be after the cropping. This is supposed to be a sequence of the GAA-TTC sequence is which is found on the desired on the selected species, and the foreign DNA from here, that foreign DNA may be amalgamated to this species and the recombination maybe takes place. This recombination is based on the desirable characters. So what will be the desirable character? Let us take an example of the crop. The crop must be resistant. It has to resist to the insects in very limited span of the times, means in a very short time the crop has to be mature. It must give a great or in a large quantity of the production. These are the desirable characters. Scientists look to those positive or those important characters and they work on that. This is the biotechnology, and that was all about the biotechnology. And this is a Monsanto, that is a Mon 100 and 810, contains a gene obtained from a Bacillus thuringiensis of the soil bacterium. The scientist has been selected the Bacillus thuringiensis, and which produces a protein protecting against the corn borer, or not only corn borer, the other cotton ball was also protected by this protein protecting or the Bacillus thuringiensis soil bacterium's enzymes may be resistant to the other foreign attack on the cotton and other these 10 genetically modified crops. Thank you.